Well, hello, everyone. My name is Greg. I'm a postdoc in the imaging platform at the Broad Institute, and I'm really excited to be here today to tell you about some science. It's this project, Predicting Cell Health Phenotypes Using Image-Based Morphology Profiling, I've been working on over the past year and a half or so. But I really inherited this project, and the project was started much earlier on, and really I was lucky enough to just take it to the finish line. And so these are the scientists that have contributed: Masha, Sukasa, Stanley, Tim, Bill, Anne, Paquita, Shantanu, William, and Federica. And so let's just jump in. So we have this phenotype, the specific phenotype that we're interested in here, which is the number of gamma H2AX spots in G1 cells, or the amount of DNA damage that exists in cells that are in the G1 cell cycle stage. And so these points here represent individual perturbations from three different cell lines, A549, ES2, and HCC44. And R squared represents the performance of the model. So when I say performance, I mean comparing the predicted output from the model to the ground truth readouts. And so in our framework, we have a test set. So this is 15% of the data that was randomly held out. So the models have never seen these readouts before. And a training set, which is 85% of the data. And we know that this is somewhat moderate performance, 0.51, but still detecting signal, because after a permutation approach, we still see training performance after randomly shuffling the features, but then the test set performance kind of falls flat. So this is just one model. And now a separate view can look like this, where we see the width of the bar here represents 0.51, where this is 0, and then this is 0.5. And each of these 3 points represents an individual cell line performance. So to expand this out, we see actually through our approach, we're making cell health readouts, we're actually making 70 different ones. So not just the number of gamma H2AX spots in G1 cells, but also several others that we're listing here on the y-axis. The x-axis is R squared, where the higher the bar, the wider the bar, the better the performance. And so where they're also ranked based on high to low. And so there's a handful of models that are performing really well. So the live width to length ratio, predicting the number of live cells, predicting the percentage of dead-only cells or live cell roundness, the number of cells in S phase. And there's other models that are performing to a moderate degree. Just in general, the number of gamma H2AX spots, the number of cells in mitosis, reactive oxygen species, apoptotic cells, and so on. We do see quite a lot of cell line variation, but in general, we are detecting some signal. And so to go into a little bit more detail about how we're actually making these measurements, we're doing this through a series of assays we're calling the cell health assays. And so the cell health assays are a series of 2 different panels. The first is the cell cycle panel, in which we measure 4 different reagents, EDU, pH3, gamma H2AX, and HUXT. And so we use the HUXT to identify cell nuclei, so basically as a cell segmentation, a way to segment cells. And then EDU, pH3, and gamma H2AX to measure various different aspects of each of these single cells. For instance, we use EDU and pH3 to identify cells that are in various cell cycle stages of G1, S, G2, or mitotic cells. And then within each of these cell subpopulations, we count the number of gamma H2AX spots per cell subpopulation. And then in the second panel of the cell health assays is the viability panel. And so in this panel, we measure 3 different targeted reagents plus Brightfield. The first is caspase 3.7 to mark apoptotic cells, DRAC7 to measure dead cells, and CellRox to quantify the reactive oxygen species that are present. And just in these small examples, it's worth noting how much variety we're actually seeing. Like this specific cell is just exploding with DNA damage in yellow. And you see a bunch of these individual cells are either already dead or actively dying. So it's really interesting to see the heterogeneity. And so really, the cell health assays they consist of these series of semi-automatic gating strategies and relatively expensive reagents, and it's not necessarily going to scale all that well. really what we want to see is if this information is embedded within cell painting images. So cell painting is an unbiased assay that isn't targeted to any specific biology. It's just kind of there to measure generally cell state. And so in the cell painting assay, we measure various different compartments: the nucleus, the ER, nucleoli or cytoplasmic RNA, AGP, and mitochondria. And so using these unbiased dyes, our task was to see if we could predict the actual cell health readouts. But to go from here to actual data for the models, we performed a profiling pipeline to get individual readouts, morphology readouts from perturbation profiles. So this means, collecting the single cell readouts, processing them, normalizing them, and generating consensus profiles, which are specific profiles per perturbation. This enables us to set up this machine learning framework in which we use our cell painting images. So these are the consensus profile morphology readouts to predict each of the 70 cell health readouts that are derived from the cell health assays independently using 70 different regression models. And so really what it boils down to is to fit a model to identify model coefficients using the cell painting measurements to predict each of the cell health phenotypes. And so a little bit more on the framework so that we can understand some model limitations and some of the limitations of our findings. We have matched perturbations, which enable this approach. So we have the same exact perturbations in cell painting as we do in the cell health assays. So in cell painting, we measure 952 different morphology features, and in cell health, we measure 70 different phenotypes. The perturbations that we selected were based on CRISPR. So these are CRISPR knockouts. We targeted 59 different cancer genes that we selected because they're known to be important cancer genes and also known to induce various morphology states. And we designed about 2 guides per gene, which came to 119 guides total measured in the 2 assays. That's our fundamental perturbation as a CRISPR guide. And then we also applied all of these 119 guides to 3 different cell lines, A549, ES2, and HCC44. So through the procedure, because there's actually no fundamental way to link the single cells between the 2 assays, right now, they're independent assays. So that means each single cell, the common unit of perturbation is going to be the guide, the consensus profile. So you can't even match replicates to one another. So through this approach, we actually only end up having 357 different samples. So that's a really small data set. It's just the purpose was a pilot study to see if we can extract signal at all from this approach. And so that didn't warrant applying any really, heavy duty approaches like deep learning just yet. We just kind of wanted to see a signal existed. So we used the most simple models that you can think, and that's an ElasticNet logistic regression model that, like I mentioned, are independently optimized and evaluated with R squared. And so why do we even think that this type of approach is worthwhile, and how is it useful? So it turns out that labeling cells with ground truth is really tough. Almost always it requires some form of manual annotation. So you have to kind of steal away a cell biologist who already have literally no time to do. And so with our cell health assays, we can, even though it's semi-automatic and doesn't necessarily scale all that well with expensive reagents and whatnot, it still is better than manual annotation. And also in general, cells are heterogeneous mixtures. So even cell lines that are plated in a dish, they're actively dividing and in different cell cycle stages and various different cell states. So it's worthwhile to see if we can identify subpopulations within those states to perhaps get some targeted profiling done. And even in more complicated mixtures, like in a patient line or some patient tissue or patient sample, perhaps we can identify disease versus healthy cells, and then do targeted profiling that way. In general, also, it's this type of approach is just going to, in general, diversify our understanding of cell state and tell us some mechanisms. And so a really easy example of this is to just think about cell death. And so it turns out that there's many different ways that a cell dies. There's many death pathways. It's not just a binary yes, no, dead, alive. But the typical technologies that do this in high throughput, like Achilles and Prism, really all they're doing is counting cells. So how many cells are present before or after perturbation. But we do know that cell death has been classically defined based on morphology. So that signal exists. Cell biologists have identified that signal. It's just a matter of developing approaches to extract it in a high throughput way. So really the holy grail here is to see, we don't want to have to collect cell health assays for every single cell painting data set that comes our way. We want to see if our models that we've trained on one small data set can generalize to a larger data set. So that's exactly what we've done here. We have a legacy cell painting data set that we've collected a couple of years ago from the Drug Repurposing Hub. So these are compounds that are candidates to be repurposed for other diseases. So this is a relatively large data set where we have about 1,500 perturbations in different doses across at least 5 replicates. And so in the paper, we present a couple of anecdotes validating the generalizability of the transfer learning between the CRISPR perturbations and the compound perturbations, and this is just one of them. But here we show the cell health models that were trained in CRISPR and applied to the Drug Repurposing Hub, the predicted amount of reactive oxygen species on the y-axis, and then the predicted G1 cell count on the x. The points that are colored in red here are the controls within the Drug Repurposing Hub. So these are bortezomib MG132. These are 2 proteasome inhibitors, and then in here, these are the DMSO negative controls. The proteasome inhibitors are known to induce ROS, so it's nice to see that they have high predictions, whereas the PLK inhibitors are actually known to restrict entry into G1. So it's really nice to see that they are not having any predicted ROS, but they actually kill cells this way. And so we see a really nice dose curve with one of these compounds, HMN214, validating our predicted G1 cell count model. And so because there's so many different compounds and many different cell health models, we've produced this web app to peruse at your leisure the various predictions across all of the compounds in the cell painting set, or the cell drug repurposing set. And so we've also listed out several resources, which I'll include in the description, of various open science resources that were generated as a result of this project, including the preprint, various different forms of data, the software, and as well as the web app. And so with that, thank you for your time and attention, and I'm looking forward to any questions. Thanks.